Alright, bro. So this is part 3 of polysaccharides. So in the last part, we covered water potential and its kind of importance, its role. Now, the first part we covered starch. So we went through amylose. Now, amylose is like a coiled structure with 1 to 4 glycosidic bonds. And then we had amylopectin, which is like a branched structure, a very branched structure that has 1 to 4 and 1 to 6 glycosidic bonds. So we looked at our boy alpha glucose and what he can do, or she, I don't know. Probably he. Now we're going to look at beta glucose. So beta glucose, what does it do? So we put these two fellas together and we want to react to them because these guys can form polysaccharides, but it's a little bit different. So we put them together and some of you absolute nerds might have figured out a problem, and there's a little problem here. What is it, bro? Well, the problem is when you line up these two beta glucose molecules together, you can't really form a glycosidic bond. Why? Because the hydroxyl group, the hydroxyl groups are not adjacent to each other. So they can't react together and they can't form a glycosidic bond and release your water. Big problem. But there is a way around this. And the way around this is a little bit silly, but basically the second beta glucose does a bit of a backflip, right? He does a backflip, he spins around, and when he turns around, hydroxyl groups are now adjacent to each other. As you can see, they're level with each other, they're adjacent, and they can react. So it's very silly, but it's basically, that's how it happens. That's how beta glucose forms its polysaccharide, its massive chain. Unlike his cooler, swaggier, badass cousin alpha glucose, they can just react adjacently without needing to turn, without needing to flip. And so that is something you need to learn, bro. So beta glucose, they're level with each other now, and they could just go ahead and form their glycosidic bond. They release a molecule of water, and another beta glucose can just come in and react normally. Because again, the second one is already flipped, so the hydroxyl group is level with each other here. So they do it again, just like before. They come together, they react. And if we zoom out just a tad for some space, you can see this little trisaccharide of beta glucose that has just formed. Now another beta glucose comes in and boom, you are faced with the same issue again. So because the second glucose is flipped, but the third one isn't, now you have the same issue where the glycosidic bond can't be formed because the hydroxyl group is not planar, it is not adjacent to each other. So what happens, bro? It just flips, okay? It does another backflip, it flips. And that way, the hydroxyl groups are now adjacent to each other again. So they form their molecule of water, they get their glycosidic bond in, and they live happily ever after. So this is how beta glucose works. You can see a trend here, a little pattern. Basically, and you need to learn this for your exam, when a beta glucose molecule wants to form a polysaccharide, every second molecule is flipped. And you can see it here, look, the first molecule, the 6th carbon is pointing upwards. The second one, the 6th carbon is pointing downwards. Third one, 6th carbon is pointing upwards. The fourth one, 6th carbon is pointing downwards. And so they can keep reacting with each other, right? They can keep adding on and on and on to form these layers and layers. And they form their polysaccharide, right? And what you should know is, kind of like amylose, they also form hydrogen bonds between each layer. But instead, these guys don't coil. They're a straight chain. So they're very straight. And this whole structure, we kind of call it a microfibril. And when it all kind of comes together, we call it cellulose. And that's what makes cellulose so special, bro. It's that all these little hydrogen bonds in between each layer of beta glucose molecules. These hydrogen bonds, so many of them form, and it makes it really strong. It's a very strong structure. And just like amylose and amylopectin of starch, this is also used by plants, but not for energy. The plant doesn't use this for energy because why would it need to use it for? It doesn't use it for energy. It uses it for protection. Right? Because this massive molecule is so strong, right? Think of it as like a bodyguard for the plant. It has such a high quantity of hydrogen bonds between each layer that it's so strong that it protects the cell, the plant cell. And that's the thing, bro. This cellulose molecule is found in the cell wall of plants. And this does go into cell biology and all that bollocks, which is something we'll cover soon. But for your biochemistry, bro, just understand that this cellulose molecule is really strong, so many hydrogen bonds, and it protects the cell from damage, such as, water pressure or all this bollocks, right? It's really strong. So learn that, bro. Straight chain beta-glucose polymer, really strong, protect the cell. And bro, don't you dare say it's found in animal cells, because then you're just finished. It's over. So in summary, bro, starch and cellulose, they are both found in plants only, okay? Now to get a bit silly here, this isn't like part of your spec or anything, but if you was to get cellulose and grab a nitronium ion, so NO2, and if you were to replace one of the hydroxyl groups on cellulose with nitronium, what you'd create is actually an explosive, right? A very dangerous chemical, flammable, and it releases toxic gas, an explosive. It's called gun cotton. It's pretty cool. Not part of your spec. You don't actually have to know this. It's just having a bit of a giggle here. And don't try this at home or anything, bro. Like, don't mail this to anyone. That'd be like a dickhead move. But it's just for your own educational purposes. I'm just having a bit of a giggle. Alright, bro. So here we have our boy, and you should know who this is. Can you name him? This man is named amylopectin, right? Part of starch. Again, we did speak about this in the last episode? Video? Whatever. This is amylopectin, 1 to 4 and 1 to 6 glycosidic bonds, used for energy. Branch structure. So what if I was to tell you, bro, that this guy, he's a jealous individual, okay? Very jealous man. The reason why is because he has like a cooler, badass cousin, right? And he's very jealous of this cousin because he's better than him in every way. Like, this cousin has it all. He gets the girls, he's cooler, and amylopectin wishes he was like him, but he isn't. Now, this cousin is called glycogen. And again, they are similar, right? They're both branched structures. They both have 1 to 4 and 1 to 6 glycosidic bonds. They're both alpha glucose polymers. They are both carbohydrates. They are both used for energy. Except there are 2 key differences, okay? Glycogen, again, is cooler. Why? Because he is more branched. Way more branched. He is so much more branched than puny amylopectin. What a pussy. And this is due to its adaptation, which leads us into a second difference, which is glycogen is used in animal cells instead of plant cells, okay? So glycogen is way more branched, and the reason why is because it needs to break down quicker. So they're broken down much faster, which means they release energy much faster. And this checks out, because if you think about it, right? A plant, he just sits there, he does nothing all day, right? He's just chilling, he doesn't move much, he's just chilling, right? If you've ever spoken to him before, they're really calm. Whereas an animal, they are obviously moving around, they're running away from predators, they're doing backflips, whatever they're doing, right? They're moving around constantly, which means they have a higher metabolic demand, okay? They're using up energy much faster, and due to that, they need a better carbohydrate storage molecule, right? They need a better way of releasing energy, and this is where we introduce its cooler cousin, glycogen. Glycogen is much better, more efficient, because it can break down easier. Like I said with the enzymes act on free ends, and because glycogen has more free ends, it can break down much faster, so it can release energy much better than amylopectin. So bro, learn that. But other than that, they are very similar, right? They're both insoluble, all that bollocks. Alright bro, so you're going to need to know how to test for them. So if someone gives you a random sample and they say, yo bro, is starch inside this? You're going to need to know how to test for it. And it's pretty easy, bro. This biochemical test is called the iodine test. And it's very easy. All you do is you grab iodine, dissolve it in potassium iodide. And the thing about this solution, potassium iodide, is it's browny orange. Due to the iodine, it's browny orange in nature. And so I grab my unknown test tube and I put my iodine solution inside. Now, if there is no starch, it will remain its browny orange color, okay? Now, if there is starch, it will turn to a blue-purple color, and it happens instantly. And that's all you gotta know, bro. Now, let's finish this video with a little cheeky exam question, okay? It says here, yo, bro, starch molecules and cellulose molecules have different functions in plant cells. In it, my G. Each molecule is adapted for its function, bro. Explain one way in which starch molecules are adapted for their function in plant cells. And this one is for 2 cheeky marks. Alright, bro, lock in and get this one done yourself. So instantly by reading the question, you should know that it commands you to give an explain, right? It's an explain question. So why? It wants you to know why. So tell me and then explain why this happens. So for an answer, we did go through this. For this answer, we can say starch is insoluble. Why? Why is this important? Well, it doesn't affect the water potential, therefore there's no osmosis. You could also say another way it's adapted is, starch has amylose, and amylose is helical, which is a very compact structure, meaning it can contain so many molecules of glucose, which, when broken down, releases energy. You could also talk about its boy amylopectin and how it's a branched structure. And why is this important? Well, again, branched structure, very many free ends, and it can be broken down easily, okay? And so, in this type of question, when it asks you to explain something, okay, you get 1 mark for telling them what it is, and then you get the 2nd mark for the why, okay? That's the most important part. Explain why. So when it says explain, say why. So if you said any one of these points with the why, you get yourself 2 marks, bro. Come on, man. Easy. Alright, bro, that's about it. Make sure to subscribe the video, all that bollocks. We're probably going to be doing amino acids next, so yeah, I'll see you soon, bro.